This video will demonstrate the steps required to design and order all necessary reagents for adding a fluorescent tag to a protein using the Invitrogen TrueDesign Genome Editor. After logging into Thermo Fisher Connect and accessing TrueDesign Genome Editor, you have several experimental options to choose from. For this workflow, we will choose gene tagging and click Next. On the next screen, you will see 5 species to choose from. Select the host organism for your gene tagging experiment and click Next. You will then select gene symbol or Entree ID as the input for how you will identify your gene of interest. If you want to target non-transcribed regions, you can use the DNA sequence or the chromosome locus option. Click Next. When you begin typing in the gene identifier box, a filtered drop-down list will appear. Select your gene of interest from the list and click Search Gene. The next screen will display all the protein coding transcripts for your selected gene. Please note that the principal transcript for the gene is displayed at the top and is selected by default. If there is a different transcript you wish to target, select that. You can easily click the gene symbol hyperlink to be taken to the NCBI website where you can visualize the transcript maps. When you have made a selection, click Next to proceed to the sequence editor view. The sequence editor will update to show the entire transcript's topology along the top of the screen with a zoomed-in sequence level view below. TrueDesign Genome Editor also displays genetic variants present across populations in the gray bar above the DNA sequence. Click on the genetic variant nucleotide to view its details, such as the frequency and genomic position. It is important to pay attention to genetic variants as they can affect the efficiency and specificity of guide RNAs. To view variants in a specific population, click on the drop-down on the right. The type of tag can be selected based on the level of expression of your gene. For a gene that is expressed, you can select a fluorescent tag or an epitope tag. Once the type of tag is selected, select whether you want your tag to be on the N or C terminus of your transcript, and the view will update to show the region of insertion. Non-expressed genes can only be tagged on the C terminus. Next, select various fluorescent proteins or epitope tags based on your previous selection. Blastocidin or puromycin can then be selected for a resistance marker. When your selections are complete, click Add Tag. The sequence editor display will update to indicate where the tag will be inserted. If you wish to change any of the parameters, you may do so at this time by clicking on the blue undo arrow under Actions in the Edit list. If everything looks right, click Next. This step might take a few minutes while the software searches the region for available PAM sites and assesses the guide RNA designs for efficiency and specificity. When complete, you will see a table with 2 tabs, CRISPR and TALEN targets. Each row in the CRISPR table displays a CRISPR guide RNA design, its PAM site, its weighted score for efficiency and specificity, the number of predicted off-targets in the reference genome, the edit site from the tag insertion site, and the primer sequences required to add the homology arms to the donor template provided in the TrueTag donor DNA kit. If you wish to find off-targets in the variant genome selected on the previous page, click on Search in the Off-Targets in Variant Genome column. This search will take an additional 70 to 90 seconds. Green checkboxes indicate recommended guide RNAs due to score and proximity to the insertion site. Visualize the guides on the sequence editor display by clicking anywhere in the row. Genetic variants are indicated below the guide RNA and PAM sequence to easily identify guides whose activity might be affected. Click on the number in the off-targets in the reference genome column to view more details such as location, the probability of cutting at the off-target site, and annotations of whether the off-target site is in the exon or an intron of a gene. Click on the number in the off-targets in the variant genome column to view more details about the off-target sites in the variant genome as compared to the reference genome. Off-target sites that have higher off-target activity in the variant genome are flagged by an orange warning symbol. To view the TALEN targets, click on the TALEN tab of the table, and similar information will be displayed for each TALEN pair. To select one or more guides or talents to add to your experiment, use the checkboxes in the table and click Next. The summary page will display all of the reagents needed for your gene tagging experiment and give you the opportunity to add additional products to complete your workflow. For a gene tagging experiment, the TrueTag verification primers, the TrueTag donor DNA kit, the guide RNA, and the TrueTag donor primers designed by TrueDesign Genome Editor are selected by default and cannot be unselected. Other required reagents include Cas9, which can be ordered as an mRNA or protein, transfection reagent, and sequencing primers for verification of the edit. Lastly, you may add positive and negative experimental controls. When you have completed your product selections, click on the Download Designs and Protocol to download your entire experimental design for future use before clicking on Add to Cart for easy one-step ordering of all selected reagents.